On this first lecture, we are going to talk about microbiology as a science. We will stop briefly on the history of microbiology first. Then we will talk about different types of microorganisms and their main characteristics. And after that, we will talk about how we create names for microorganisms and what rules we have to follow when we create a new name for the new microorganism. In the second part of this lecture, we will talk about bacteria, their characteristics, once again in more details, their structure, and requirements for growth. So, microbiology is the study of microorganisms. So what do we call microorganisms? Microorganisms are living things that are too small to be seen without microscope. The first microorganisms were discovered back in the 1600s. In 1665, Robert Hooke reported that living things were composed of little boxes. Today we call them cells. In 1858, Rudolf Virchow said cells arise from pre-existing cells. And today those two statements form what we call cell theory. And according to the cell theory, all living things are composed of cells and come from pre-existing cells. Another name of scientist I would like you to remember is Anthony Leeuwenhoek. He was the scientist who created the first microscope. With the help of that simple microscope, he was able to observe microorganisms. He noticed that they were everywhere. Also, he noticed that they could move. That means they were alive. Their numbers increased rapidly. That means they could replicate. Also, he noticed that microorganisms always appeared suddenly in certain materials that had previously been free of them. For example, nutrient broth or plant extract. So it seemed logical back then that microorganisms were products of spontaneous generation. So besides Anthony Leeuwenhoek, a lot of other scientists back in 1600s believed in spontaneous generation theory. So what is it? According to spontaneous generation theory, all living organisms can arise from non-living matter. And of course there was an alternative theory back then. It's called biogenesis. And according to biogenesis, all living organisms arise from pre-existing life. Because there was an opposite theory to spontaneous generation, we call it biogenesis. A lot of scientists believed in biogenesis back then, and they tried to disprove spontaneous generation theory. One of them was Francesco Redi, Italian physician. He did a very simple experiment with jars of meat. He took 6 jars, placed decaying meat on the bottom of each jar. Then he covered 3 jars with fine net, and 3 jars he left open. A few days later, maggots appeared in the surface of meat of uncovered jars. So where did the maggots come from? His conclusion was that maggots developed only on the meat that flies could reach to lay eggs on. But critics were not convinced, and they said their conclusion was maybe it worked for maggots, but microorganisms were exception of this rule. For 80 years, the debate continued. John Needham, by the way, believed in theory of spontaneous generation and tried to prove it. For his experiment, he took chicken broth, boiled it, because everyone knew boiling killed microorganisms. Then he placed boiled chicken broth into a flask and sealed it. Few days later, broth became cloudy, meant something was growing there. So his conclusion was microorganisms could develop in that flask only by spontaneous generation. But Italian priest and professor Lazaro Spallanzani was not convinced. He said perhaps microorganisms were in a flask or entered broth after boiling but before sealing. So he decided to perform the same experiment but in different order. He took chicken broth, placed it in a flask, sealed it, and then boiled it. A few days later, the broth was still clear, means no growth. But still critics were not convinced. They said it didn't disprove spontaneous generation. It just proved that spontaneous generation required air or oxygen. For another 100 years, microbiology didn't develop as a science until 1859, when the French Academy of Science sponsored competition to prove or disprove theory of spontaneous generation. And this is when young French chemist entered the stage. His name was Louis Pasteur. He performed very simple but very elegant experiment and was able to disprove spontaneous generation. So what he did was he took flask, placed nutrient broth into that flask, then he heated the neck of that flask, pulled it and curved it. After that he boiled nutrient broth in the flask, and a few days later broth was still clear, means no microbial growth. Then he tilted the flask so some broth entered the neck and then tilted it back so broth returned to the flask. A few days later, broth became cloudy with bacterial growth. So the main idea of his experiment was by pulling and curving that neck of the flask, he created barrier for microorganisms, because from that curve microorganisms couldn't get into the broth, but air could freely go in and out of flask. But you have to remember that Louis Pasteur was a very talented scientist, but at the same time he was a very lucky one. For his experiment he took meat broth. If he took plant broth, he would fail. Why? Because plant broth usually contains a lot of what we call endospores. Endospores are the most stable biological structures. Some bacteria are able to form endospores when environment is not favorable. Let's say it's too hot or too cold for them to grow, or there's not enough of nutrients. Then bacteria cover themselves with endospore, protective blanket, and stay in this resting condition as long as they need. When environment becomes favorable again, they remove endospore and move into what we call vegetative stage. Please remember, vegetative stage for microorganism means actively growing stage. After spontaneous generation was disproved, golden age of microbiology began. Uh, As I said, Louis Pasteur was a very talented scientist, and besides the fact that he disproved a theory of spontaneous generation, he also discovered relationship between microorganisms and infectious diseases. He discovered immunity, and the first vaccines were created by Louis Pasteur. He developed first antimicrobial drugs, antibacterial drugs. Some of Louis Pasteur's discoveries we still using today. For example, fermentation. Louis Pasteur discovered that certain microorganisms can convert sugar into alcohol, and this is how we make beer and wine today. He also noticed that certain bacteria can ferment alcohol into vinegar, and this is how we make vinegar today. Another discovery that we use widely today is pasteurization. We use pasteurization for dairy products. Pasteurization is a special heat treatment used to control microorganisms in milk and dairy products. Guys, please remember, pasteurization is not sterilization. We do not boil milk and dairy products. We just expose them to high temperatures for about 70 degrees of Celsius for about 15-20 minutes. This kills most pathogens in that milk. It also kills lactic acid bacteria that cause spoiling of the milk. Another very talented scientist from Germany I would like you to remember is Robert Koch. Remember I told you Louis Pasteur discovered relationship between microorganisms and infectious diseases. He proved that microorganisms cause infections. Robert Koch made one step further. He actually proved that only certain bacteria can cause specific symptoms of certain infectious disease. Today, his experiment known as Koch's postulates. He studied anthrax, disease or infection that can infect animals and humans. First, he noticed that the same microorganism present in all blood samples of infected animals, and those animals develop symptoms of this infection. The second step, he was able to cultivate, to grow these microorganisms, and today we know this organism as Bacillus anthracis. It causes anthrax. Next step, he injected healthy animals with this organism, and the last step, he observed animals became infected and developing symptoms of anthrax. So this way he proved that only Bacillus anthracis can cause symptoms of anthrax. Next, we're going to talk about how we create new names for microorganisms and what rules we have to follow to create a new name for the new microorganism. So if you look at the bottom of this slide, you see that there's a name of bacterium, Escherichia coli, and this name is composed of two words. The first word, this is what we call genus name, and as you see, it is always capitalized. The second word is called species name or specific epithet, and it is always written in the lower case. All names of microorganisms must be italicized or underlined. Also, you can use a short version of genus name. So instead of Escherichia coli, you can say E. coli, and that would be correct. Next, all names are Latinized. That means all those words that we use to create names came from Latin. And that means Escherichia coli or E. coli is called E. coli here in the United States. The same in Japan, Germany, French. So it means it's used worldwide. Also, some microorganisms might have two names. For example, Mycobacterium tuberculosis. This is a name of bacterium that causes tuberculosis in humans. But you have to remember that Mycobacterium tuberculosis is also very well known as Koch's bacillus, because Robert Koch was the first who discovered and described this bacterium, and actually he received Nobel Prize for that discovery.